everyone. Today we are going to continue our conversation about shunts off of the glycolysis pathway, and we'll be talking about a specific shunt called the pentose phosphate pathway, and then we'll be talking about the products of that and why it's an important shunt to use and when it is important to use it. So the pentose phosphate pathway branches off of glycolysis right at the beginning. So it branches off at step 1, which is where you have glucose 6-phosphate. So the only thing on top of that would be glucose at the very beginning of glycolysis. And then you convert that into glucose 6-phosphate. And pretty much right off the bat, this is when you begin the entire pentose phosphate shunt. Now this pentose phosphate shunt is really important for providing something called NADPH. So we've seen NAD, we've seen NADH, but now we're looking for NADPH, and this will become a precursor to things like DNA and RNA. It's important for making those things. It's important for making fatty acids and some amino acids too, as well as something called reduced glutathione, which we're going to get into at the end of this video. That is an antioxidant. It's a very important antioxidant within your cells. So this is kind of the main thing that we are looking for in the pentose phosphate pathway as we leave our glycolysis pathway, do all this stuff, and then we can re-enter the glycolysis pathway again. Now where this shunt re-enters the glycolysis pathway is in two places, at the 6P or fructose 6-phosphate, so fructose 6P over here, or when we get into the glyceraldehyde 3-phosphate farther down, just step 4 in glycolysis. So it's got two places where it can re-enter glycolysis. Now this map is just kind of showing everything, the whole pathway and the various branches we can take and all the different routes we can go. If you notice, a lot of these arrows are reversible, so many reactions are of course reversible. These are other examples of reversible reactions. Now when we see arrows like this, so we've got two reversible arrows kind of leaning into each other like that, what we're seeing are two products that are dependent on each other. They're going together to create new products, and they can go the other way too because it's reversible. If the arrow was only going one way, then they would be going like that. It's sort of like when we've seen different reactions. So we'll just say product X and product Y, and then we have, say, NAD becoming NADH. This little thing that we've seen, this sort of offshoot, the byproduct almost of the reaction. So this is the substrate, this would be the product, and this is something that's happening off to the side, dependent on the reaction, but not directly involved with either the substrate or the product. These little arrows here, like this, that's what this is doing. It just looks a little bit funny because we've got two of them and they are reversible, so it's just kind of added a little bit onto this little more simple schematic there. Now this picture can also be represented as this picture, which I think this picture, although when I first flipped to it, I was about to say it's easier to look at, but okay, maybe it's not. But when you really get into it, it will become easier to look at. So I'm going to work with this picture. It has all the same things on it, so just arranged a little bit different. Like when you're looking for 3-glucose 6-phosphate, we're going to find that right there. When we're looking for fructose 6-phosphate, that would be the where we can re-enter the pathway. of glycolysis. There's glyceraldehyde 3-phosphate there. That is right there. So it has all the same parts on it, but they're kind of spread out a little bit more, I think, rather than all jumbled together. So we can follow along with it a bit easier, I think. So let's just start up here. We'll start at the top and we'll just kind of work our way around. So we've got our glycolysis pathway here. Let me use a different color so I don't confuse. So there's glycolysis. If we were to continue, we would just keep going with glycolysis and finish it and end up with pyruvate and on into the Krebs cycle. But we're not worrying about that. We are taking a shunt. We are leaving the pathway right here at the glucose 6-phosphate. Now this, don't worry about this part right here. Tried to cut that out because we're not there yet. But we're just looking at what's happening to the glucose 6-phosphate right from the start and why it's going to go into this pentose phosphate pathway rather than finish doing glycolysis. A big part of the reason of why it would do that would be this enzyme right here, this G6PD. That's glucose 6-phosphate dehydrogenase. We know what a dehydrogenase is. It takes a hydrogen off of something, in this case glucose 6-phosphate, and it's going to be using that hydrogen to reduce NADP. So this gets reduced, and NADP is just NAD with a phosphate added to it. But when we reduce it, oops, I'm sorry, yep, we end up, I'm sorry, I lost my train of thought for a second. When we reduce it, we get NADPH. So this is reduced into this. And this is what we were looking for. This NADPH can go into fatty acid synthesis, among other things. Well, we're going to keep following it along as we go through the pathway. Now our product, after we took the hydrogen off of our glucose 6-phosphate, is this 6-phosphogluconolactone. Now we're going to work on that with another enzyme here. It says 6-PGL. That is 6-phosphoglucanase. Again, just named right after the thing that it's working on. We're going to be taking water here and breaking it apart in this reaction. So we're going to get hydrogens as a byproduct of this reaction. There's those little curved lines there showing kind of an offshoot of this reaction. And we end up with 6-phosphoglucanate. Now here we get the same enzyme working again, the glucose 6-phosphate dehydrogenase, and it's going to again be oxidizing and reducing. It's removing a hydrogen from this, putting that hydrogen again onto NADP, or NADP, giving us NADPH. So again, it's reducing NADP into NADPH. So there's another source of NADPH that can go into fatty acid synthesis. Okay, now what this leaves us with is ribulose 5-phosphate. Now this is the end of the first stage of the pentose 5 pathway, or the pentose, not the pentose 5 pathway, the pentose phosphate pathway. This is called the oxidative phase. So where it says PPP here, that just means pentose phosphate pathway. So this is the oxidative part of the pentose phosphate pathway. And it's called the oxidative part because of this enzyme, the glucose 6-phosphate dehydrogenase, is oxidizing things, glucose 6-phosphate and 6-phosphoglucanate. It's oxidizing those, taking the hydrogen away, and we're using that hydrogen to reduce NADP to make NADPH. Okay, so that's why we call it the oxidative part. But we're not done yet. Even though we've got our NADPH, we can get other things out of this pathway too, really important things. Things that will enable us to not just make fatty acids, but we'll also be able to make amino acids. We'll be able to make our nucleic enzymes, like not our nucleic enzymes, our nucleotides, so things like DNA and RNA. And all of that will happen in the non-oxidative phase of the pentose phosphate pathway. Now, so we have two ways that this can go, and it'll go simultaneously, depending on the various amounts of these enzymes here. Now, this enzyme, RPE, this is called a Ribose 5-phosphate 3 epimerase. We don't need to think about what an epimerase is exactly. It just kind of flips component parts of a molecule from one side of the molecule to the other. That's what it does. We're changing a molecule. Kind of similar to that is this one, RPI. It's Ribose 5-phosphate isomerase. An isomer is a rearranged molecule in a different kind of way, rather than flipped. It's just, we've rearranged the actual order of the molecule. So it's just changing the structure of a molecule without changing the chemical formula of the molecule. Both of these enzymes are doing that. Now, let's follow RPI first, so the isomerase. We'll just follow down through the products here. Don't worry, you don't have to memorize, of course, these names of all these things that we're going over, but just to be able to follow along with this and to be able to interpret this metabolic map that we have here, that if I were to say, we were doing the pentose phosphate pathway, and we had more RPI and let's just say no RPE, what are we going to do? And you would be able to follow it down, okay, everything over here, everything distal to where we introduce these enzymes, everything distal would be going in this direction rather than this direction. And I'll go over that part again as we get into how we regulate this pathway in a minute. But when we're doing RPI, when we're using RPI, we're going to go this way. And this is how we can end up making our DNA and RNA going through this particular pathway. Now this will also continue on down here, fructose 6-phosphate. If that sounds familiar a little bit, it's because there it is, fructose 6-phosphate. That is re-entering glycolysis. So that would be how that part re-enters glycolysis. Now let's follow the epimerase instead. So let's just ignore that and we'll follow the epimerase. And that would go this way. So there's two products of note here as we follow these two down. Let's look at this one, the erythrose 4-phosphate. The erythrose 4-phosphate goes into amino acid synthesis. Not all amino acids, just some amino acids. So this isn't the main way that we create our amino acids in the body, but it is one way that we can make some amino acids in the body. But this molecule here, this part that is along the epimerase pathway, glyceraldehyde 3-phosphate, like back here, that one looks familiar too. That's going back into glycolysis. That's one of the steps of glycolysis. So here we can go re-enter glycolysis again. And that would be how that pathway goes back into glycolysis. Now, where you see this enzyme here, TKT, that's listed over here because it's used over here as well as we go back that way. So it's showing how you get back into your fructose 6-phosphate or your glyceraldehyde 3-phosphate here. This is called a transketolase. It's just working on a keto body. It's taking a keto body and moving a keto body from one molecule to another. But this TKT is essential for both this pathway, the RPE pathway, and then the RPI part of the pathway. Taldo, this is an aldolase. We don't also need to worry about this, but it's just critical for the pentose phosphate pathway. We can't finish the pentose phosphate pathway without this aldolase enzyme here. Okay, so the main takeaway here, without getting bogged down in all the names of the chemicals and everything going on there, is that we have our glycolysis, right? And this is what we're doing to create energy. This is how we're beginning our energy production. But let's say we have a lot of, NADP and not a lot of NADPH. Or if we have a lot of this enzyme. This enzyme is a really important enzyme for regulating this pathway. If you have a ton of glucose 6-phosphate dehydrogenase, you are going to be more likely to be doing the pentose phosphate pathway more. So having a lot of glucose 6-phosphate dehydrogenase increases how much you do the pentose phosphate pathway. So let's say things that affect the pentose phosphate pathway. 1, glucose 6-phosphate dehydrogenase. This just makes sense. The more of this you have, so I'll say let's increase the glucose 6-phosphate dehydrogenase. That leads to increasing the pentose phosphate pathway. Having a lot of ATP, that will also increase the pentose phosphate pathway. And just think of that, why that might be. If you have a lot of ATP, it means you've been doing a lot of glycolysis, a lot of Krebs cycle, a lot of electron transport chain. You have enough energy. You can take a break from making energy and start doing these other things, kind of cellular maintenance sorts of things, if you are having excess energy in the cell. So having a lot of ATP is almost like a negative feedback for creating more ATP. It slows down the energy part of the cell and it speeds up the other parts of the cell where we're trying to make other things. Also, having a lot of NADP will increase the pentose phosphate pathway. And it does that by increasing the activity of this enzyme, the glucose 6-phosphate dehydrogenase. The NADP binds to the enzyme, it changes the shape, makes it more active. The opposite situation, if we have a lot of NADPH, that will inhibit the pentose phosphate pathway by inhibiting the enzyme glucose 6-phosphate dehydrogenase. And those two also make sense if you think about them. NADP, this is the thing that we want to turn into NADPH that we'll use to make fatty acids that will help us continue on down to the non-oxidative part of the pathway so we can make our DNA and RNA, we can make amino acids, all those things, these other things that we want to do in the cell. We need NADPH for that. So if you have NADP without the H, that's essentially a signal to this enzyme that it needs to be more active. We need to be making more NADPH. Now if we have a lot of NADPH, that's a signal to this enzyme that, hey, we've got enough NADPH here. We have enough of all these other things. Why don't you go back to the glycolysis pathway? Let's continue making energy instead because we don't need to be doing this anymore. We've done this enough. So this would be a negative feedback on its own. Okay, we've done enough of the pentose phosphate pathway. Okay, now another part of this that is going to be important from the NADPH point of view is that NADPH also helps in the creation of something called reduced glutathione. Which is an antioxidant. Antioxidants help to neutralize something called reactive oxygen species. These are abbreviated as ROS, so that's reactive oxygen species. So what is a reactive oxygen species? It is something that is created through normal metabolic processes. It is a molecule that is missing an electron on its outer valence. If you've had chemistry, most of you have probably had chemistry in some way. We don't need to get too deep into it, but let's look at the nucleus of a cell, or of an atom, sorry, not of a cell, a nucleus of an atom, and then we have these electron shells that are just kind of circling around. And let's just do a hydrogen here, just to make it easy. I don't want to be drawing a ton of electrons just to make this point. But this inner electron shell, where the electrons are kind of orbiting around the nucleus of the atom, this could hold 2 electrons. In a reactive oxygen species, it just has 1 electron. And this makes it really reactive. It doesn't want to chemically, it is driven to bind to something else to balance out its electron status here. It wants to have this outer shell, in this case it's also the inner shell, but the shell that is missing the electron, it wants to have that be full. So it's going to always be trying to react with something, and this can cause damage inside the cell, specifically to cell structures, and most damagingly, I suppose you could say, to your DNA and RNA, which can cause problems, obviously, especially as damage like that accumulates later in life. So reverse oxygen species are things that you don't want to have too much of. Like I said, you get them from normal metabolic processes, but sometimes they can get a bit overwhelming just from environmental factors, pollution, toxins, just eating foods that have a lot of metabolic activity that just give off these reactive oxygen species as metabolites. So there's a whole bunch of ways that you could kind of increase your reactive oxygen species levels, and that's not a good thing. So antioxidants are important for just changing them, stabilizing them, making them okay again. For example, hydrogen peroxide, that is a reactive oxygen species. It's damaging inside the cell, so we want to neutralize it as quickly as we can. And an antioxidant can neutralize it into water. So it can be neutralized into water. So we're just knocking a hydrogen off of it, and that makes it non-reactive. It's just water at that point. So that's kind of the way that antioxidants work. We're going to look at reduced glutathione, and in Unit 2 we're going to do a whole lot of stuff about antioxidants. We can get really into antioxidants. They're interesting because you can really apply them to your own life, and what you're eating and what you're ingesting, and all that kind of stuff. Okay, so let's move on. Okay, we already kind of talked about all this stuff. This is just another image of the pentose phosphate pathway. I'm not going to zoom in because we don't need to look at the details of this so much. We already did. But this would be the pentose phosphate pathway right there. This would be glucose. I'm trying to see, or glycolysis, right here, kind of going off to the side. Here is where the pentose phosphate pathway is entering back into glycolysis, right here at the fructose 6-phosphate, and then at the glyceraldehyde 3-phosphate. Okay, so this is just a little side pathway going on. Our pentose phosphate pathway, we're getting NADPH out of that. Okay. Here is our glycolysis happening, and then it's going to go into, oops, it's going this way, sorry, can't see my own slide, it's very small, into the Krebs cycle. So what we have here are 3 metabolic maps together, because they are all dependent on each other. They are all linked together. One is branching off of the other one and going back into it. This one down here is a result of the first one. So we can really start putting metabolic maps together like this. We can have another process for fatty acid synthesis that could kind of go between these two. We can have another one for amino acid synthesis coming off of here, and eventually we will start putting all of the maps together, but they just all feed into each other. They're all really, really interrelated. But here, this is just essentially talking about the regulation of why we would take the pentose phosphate pathway, which we already talked about. High levels of ATP, that's the only thing that I have written on here. These are the other ways that we would regulate that pathway. So we already talked about those, how we might increase it, or in this case, decrease the activity of that pathway. Okay, so let's look at our NADPH a bit more. The NADPH is a reducing agent. A reducing agent, what that means is that it can donate an electron, which we've talked about that before, I think. So, and oftentimes we're looking at just a hydrogen along with its electron. So often as hydrogen. So this NADPH, we're looking at the H there, that can be given to something in order to reduce it, to make it into something else. So we can do that for making fatty acids. That's what we have shown here. I'll get into that. Cholesterol is a specific form of fat. And then reduced glutathione, which we'll be talking about as our antioxidant that we've already mentioned a bit. But before we get into reduced glutathione, I want to talk about this little map here. Not too much detail on this, because in Unit 2 we are going to be getting into fatty acid synthesis and how we make those fatty acids and all that kind of stuff. But I want you to see the relationships between the pathways, similar to what we were talking about on the previous slide. Here we have glucose. Okay, so the pathways are always, you know, when you're looking at the map, they're always set up. Sometimes they have it starting up at the top, sometimes they have it going left to right, sometimes it's going right to left, but it's always the same pathway. Just looking at it from a different point of view, or being drawn in a different kind of way, and getting used to that and being able to identify the starting point of the pathway and the different branching out points of the pathway are important when you're looking at these maps. Important for the test too. When you start, you know, when you're taking the test and you're looking at a map and I'm asking questions about a map, you need to be able to identify the familiar points of that map so you can follow it along. So there's glucose. That's familiar. That is easy to spot. That's going to be the start of the pathway. Here they just glommed together the pentose phosphate pathway and glycolysis. There they are together. So we know that we do glycolysis, and pentose phosphate pathway can branch off of that depending on all the different things that we talked about. Now, we know that NADPH comes from the pentose phosphate pathway. And we know that NADPH is going to be important for making fatty acids. And we can follow that NADPH up here, and we get to this enzyme, fatty acid synthase, which does exactly what you think it's going to do, which is makes fatty acids. But what is it going to make fatty acids out of? We've got NADPH enabling this enzyme, which is then going to create this fatty acid here. But what we haven't done is figured out, okay, well, this enzyme is working on this. Where did this come from? Where this came from is another way that these pathways tie in together. And we need to follow it all the way around in order to see that. So let's go back to glucose glycolysis. This time we won't take the pentose phosphate shunt. We already did that. That's happening. We made our NADPH. But we're going to keep on going with pyruvate, going into the mitochondria. So we know this. We know acetyl-CoA. We know acetyl-CoA is doing the Krebs cycle. Okay, the Krebs cycle isn't really shown on here, but what we do have shown are these two things. So OAA, that's oxaloacetate. Okay, my pen is running out. Let me grab a different pen. Oxaloacetate. We know oxaloacetate is part of the Krebs cycle. It's also part of the malate aspartate shuttle, right? So oxaloacetate, there's malate. We know that they're, linked and helping to get electrons across the mitochondrial membrane. We're not worrying about the malate aspartate shuttle right now, though. We are just simply trying to identify all the little pieces they have on this diagram, and to look at this malate where this is linking into our NADPH. So let's follow the malate. We can follow up here too. We're going to first follow down here with our malate. This enzyme, malic acid, or malic enzyme, is going to be working with NADP to put us back to pyruvate. So that's a little circular thing we have here, in addition to being part of the shuttle with, oxaloacetate and aspartate. So we can put aspartate in here too. We can feed back into pyruvate and back into the Krebs cycle. So a whole other little circle here tying everything together. And part of that is linking up with this NADP and reacting with, the glucose 6-phosphate dehydrogenase that is going to take the hydrogen off of glucose 6-phosphate, put it onto the NADP and give us our NADPH. So we're right back where we started from. So let's go back up here. Let's follow our Krebs cycle. We know that citrate is part of the Krebs cycle. We know that it's the first part of the Krebs cycle, but we don't always have to be using citrate to make energy or to do the Krebs cycle. Citrate can go off and do this kind of side job that it has by being shuttled out of the mitochondria, and then going on to do this pathway. So being reconverted into acetyl-CoA, we're going to react with this acetyl-CoA carboxylase, which will give us malonyl-CoA, which is what we needed for our NADPH and our fatty acid synthase to create the fatty acid. So to summarize what we've done, we know the pentose phosphate pathway gives us NADPH. That's an important step there. We know that glycolysis can send us into the mitochondria and get us to the Krebs cycle. And we know that citrate is part of the Krebs cycle. But citrate can also end up being shuttled out of the mitochondria and ending up as part of this fatty acid production line here, all just eventually resulting in malonyl-CoA, which will react with NADPH in this fatty acid synthase to make us fatty acids. So we've got glycolysis happening over here, the pentose phosphate pathway giving us NADPH over here. We've also got pyruvate being formed from glycolysis going into Krebs cycle and citrate. And citrate can, lots of different stuff happen to it, blah blah blah, give us malonyl-CoA. And now our little pathways came together with the enzyme fatty acid synthase, and now we're making fatty acids. Okay, so that's the take-home message from this long period of time I have spent talking about this one slide, just to see how we get from our one pathway, our pentose phosphate pathway, we can influence what this other pathway is doing. Because if you think about it, if you don't do the pentose phosphate pathway and you don't make your NADPH, you're not going to be able to make your fatty acids, no matter how much malonyl-CoA you have. Also think about here the trade-off between doing glycolysis and getting it into the mitochondria, running the Krebs cycle, and using the Krebs cycle for energy and electron transport chain, versus not running glycolysis so much, doing the pentose phosphate pathway instead to get NADPH, and at the same time not using citrate for the Krebs cycle, but instead using it again to make malonyl-CoA so that you can work together with the NADPH from the pentose phosphate pathway and make fatty acids. So you're almost trading between, are you going to make energy, or are you going to make fatty acids? And just think about why, why you would want to do that. And it's, the reasons are, these things. If you have a lot of energy already, you don't need to make more energy. You can make your fatty acids. If you, already have a lot of the stuff that is required to make the fatty acids, well, then you don't need to keep on making fatty acids. You've made enough of that. So we can just look at what's going on in the cell in different conditions, and that will determine which way the reaction's going to go. If you think of it kind of like a train switch, like if you're on a metro or different train tracks, and they just flick a switch and the train can go either that way or that way or that way, and you go to a completely different place. It's like that, and those enzymes would be the train switches that switch the track for the train. Okay, so our last little bit here for our last couple minutes would be reduced glutathione. So we mentioned this. It is our antioxidant that comes from NADPH. And this picture is using these, those little curved arrows again. And remember, that just means things that are kind of coming off of a reaction. Now, what we have here, the main player, so the starting place here, would be this. This is how glutathione is existing in the cell before we reduce it. And it's abbreviated as GSSG here. That's glutathione disulfide. It has sulfide molecules on it, so sulfites, and not sulfites, sulfides, which is a slightly different, anyway, it's got sulfur in it. And there's actually 2 glutathiones in this molecule. Now with NADPH, when it comes in contact with this enzyme, glutathione reductase, it will react with the oxidized glutathione, and it will reduce it, and it will give us our glutathiones, which is abbreviated as GSH, and there will be 2 of them because there were 2 stuck into this molecule. Now, while that happened, what the glutathione reductase did to our NADPH is it took a hydrogen away and it left us with NADP, which can then go back into the pentose phosphate pathway looking for another hydrogen. Okay, so we got our reduced glutathione. This is what we wanted out of this. This slide, this should be a U here, glutathione peroxidase. So this is another enzyme that we have here. The reduced glutathione can be acted on by the glutathione peroxidase, and it will cause the reduced glutathione to react with hydrogen peroxide. The reduced glutathione is essentially going to knock a hydrogen off of the hydrogen peroxide when glutathione peroxidase is present and working, and give you water in the end. So hydrogen peroxide is H2O2. It's not knock a hydrogen off, knock a water off, or knock an oxygen off, I'm sorry. So H2O is water. So I misspoke. So we're taking an oxygen off of the hydrogen peroxide to create water. So what we've done, this is a reactive oxygen species. This is not a reactive oxygen species. So we've neutralized it. We've made it okay. We've made it so it's not going to damage the cell anymore. Now, one more thing I want to point out on this slide would be over here with our glutathione reductase and over here with our glutathione peroxidase. They both require something. What they require are cofactors. And in the case of the reductase, it's riboflavin, which is vitamin B2. And in the case of the peroxidase, it's selenium, which is a mineral that you can find in nuts primarily, or fish. So if you eat those things, you get selenium, or in a vitamin, you can get that too. Okay, so we're not going to go into a huge lot of detail on this slide. We are making this video a little bit on the long side, and we're only really kind of looking at this one side of it here. So this is another function of glutathione, and this is as an amino acid transporter. When glutathione is inside the cell, not only can it be a reducing agent and making hydrogen peroxide into water, but it can also help with getting amino acids from the blood into the cytoplasm. And what it does is it actually reacts with that amino acid out there with the help of an enzyme. And this enzyme is called gamma glutamyl transpeptidase. So that little squiggly sign is a gamma. So this enzyme, gamma glutamyl transpeptidase, is actually an enzyme that is sitting there stuck on the cell membrane. It's a membrane-bound enzyme. And that enzyme is able to grab onto the amino acid when glutathione is interacting with it. So when you have glutathione together with gamma glutamyl transpeptidase, then you can bind that amino acid and move it inside. Okay? Once you've moved that amino acid inside, it forms something called cysteinoglycine. This is a specific kind of amino acid, and it's inside the cell. And you can see that we can break it down into cysteine, we can break it down into glycine. We're not going to think about that part of it. What we're thinking about more is how it is then converted into these intermediary products, and we form glutamate. We've seen glutamate Glutamate was important as an amino acid transporter across the mitochondria in that malate aspartate shuttle. So we'll just write transports amino acids across mitochondrial membranes, both of them, the inner and the outer, in the malate aspartate shuttle. So if you think back to that malate aspartate shuttle, glutamate was used to antiport aspartate in an outward direction, out of the mitochondria, while glutamate went in to the mitochondria. So there's another little link up here. So we've gone, from glycolysis to the pentose phosphate pathway, kind of back into the Krebs cycle and the malate aspartate shuttle, and we've just linked them all together through glutathione. Okay, so that does it for this video, and our next video will be about glycogen.